Welcome to Life in Biology. I'm Dr. Joel Graff. Today we're going to go over some lecture notes from a freshman level, 100 level biology class. The topic today is nucleic acids. And to start off with, we reminded the students that we had already discussed polysaccharides or carbohydrates. We discussed lipids, and now we're on to nucleic acids. Nucleic acids and proteins are going to be kind of intertwined with each other, and that's because of the central dogma of biology. And today, when we talk about nucleic acids, we're going to focus on the building blocks, which are called nucleotides. So what is the central dogma of biology? Well, one of the co-discoverers of the structure of DNA, Francis Crick, in 1956, so just 3 years after the structure of DNA had been published, he put forth the central dogma of biology. And the idea here is to talk about the flow of genetic information. So we now know that DNA is the nucleic acid that living things use to copy and pass on to their offspring. And if you are even just a single cell organism, copying that DNA is necessary so that when you split in two, you've got DNA in both cells, both new daughter cells. Okay, so that copying is called replication. Then when DNA is, if you read DNA, an enzyme reads DNA and makes it into RNA, that process is called transcription. And then RNA can be read if it encodes a protein, and another enzyme will read that mRNA or RNA and translate it into a protein. So we've got transcription, which is going from nucleic acid language to nucleic acid language. And when you have the same language and you're just converting it from one form of communication in one language to another form of communication, it's transcription. Similar to how someone might listen to an audio recording and then transcribe the words that are being said, it's still staying in the same language. When you change languages, here we're going from nucleic acids, one of our four biological macromolecules, into protein, which is a different macromolecule. Now we're switching languages, and so then translation makes sense. So I just kind of make a point of talking about that because it's very easy, and I probably still slip up even after 20 years where I say translation when I mean transcription, and I say transcription where I mean translation. But if you're sticking in the same language, that's nucleic acid to nucleic acid, it's transcription. If you're changing your language from nucleic acid to protein, that's translating. Okay. So what are the building blocks of nucleic acids? I already mentioned it on the previous slide, but they're nucleotides. Now there's two different flavors that we can deal with. One flavor is more adequately named deoxyribonucleotide triphosphate, or and that's the building block for DNA. And then if you drop the deoxy off the front of this word, you end up with ribonucleotide triphosphate. Okay, I underlined ribo because riboses are our 5-carbon sugars. So there's going to be a 5-carbon sugar designated with an S below. Some students thought I had written a 5. I guess my S's aren't exactly proper, but my 5s look like this and the S's look like this. Is a nice zigzag. So I do make a point of making a couple different points on the 5 like that. Anyway, back to the topic at hand. Ribonucleotides, sugar-based, and then it says triphosphate. If we're dealing with DNA, there's 4 different types of deoxyribonucleotide triphosphates. You have adenosine with a triphosphate, cytosine with a triphosphate, guanosine with a triphosphate, or thymine with a triphosphate. And rather than drawing out the entire phosphate functional group, which involves 4 oxygens and a phosphate for each of those, I'm drawing it with the shorthand where you draw a P and circle it, and that's a shorthand for a phosphate. So you've got adenosine, deoxyribonucleotide called adenosine triphosphate, and so we might call that DATP for short, DCTP, DGTP, and GTTP. And if we want to just refer to a collection of these, it's DNTPs. So if you ever run across someone saying DNTPs, they mean a collection, a mixture that contains all of these. Okay, so these have triphosphates, but then when we look at the structure of when you connect all these nucleotides together, we've got one phosphate, a sugar, phosphate, sugar, making up the backbone. And then there is a nitrogenous base sticking out one side of it. So this nitrogenous base is where it's either the A, C, T, or G. Okay, so each of these, I changed some color for the first few so that it would make more sense, but each of these is roughly an L-shaped molecule, and the sugar of one molecule will be connected to the phosphate of the next nucleotide that comes along. When you connect those two together, as you add each nucleotide triphosphate, two of the phosphates go away and you're left with just one. So the two phosphate groups fall off, and that provides some energy for this reaction to happen. So again, we've got a series of L-shaped molecules, and if you use your imagination, this starts to look like half of a ladder, where this is the side of the ladder and this nitrogenous base might be sticking out to be part of the ladder. And so later on, when I draw DNA without as much detail, not that this is exactly detailed, it'll look more like just a ladder standing on its side. So here's a connection of 4 nucleotides connected together. You'll notice that one end there's a phosphate and the other end there's a sugar, and it's not drawn here, but there is a hydroxyl group that needs to be here so that the next phosphate group can attack the carbon on that sugar and connect. And so there's a phosphate on the sugar here, and we're going to talk about how it's a 5' carbon for that phosphate, and then the hydroxyl is a 3' hydroxyl. I'm bringing that up a little bit early because I don't have it drawn out yet, but just so you know, there is a direction to the DNA. So next slide, we have where I did the phosphate sugar backbone, and then I drew a bunch of L shapes in a row. Again, I pointed out that there's a phosphate. The phosphate happens to be on what's considered the 5' end of the backbone, and the hydroxyl, this is very messy writing at this point, but the hydroxyl is on the sugar, and that's at the 3' end. So what am I talking about with all this 5' and 3'? I'm going to jump over to this part of the figure next, and here is a ribose sugar. We've got 1, 2, 3, 4, 5 carbons. I didn't draw the C's for all 5 of them. I didn't draw a C here for 4 and a C here for 5, but we've got them numbered, the 5 carbons that are there. And you'll notice the phosphate group is connected to the 5' carbon, and then the hydroxyl group is connected to the 3' carbon. So if we drew an arrow pointing from the 5' end toward the 3' end, it would go in this top to bottom orientation, the way that I have this ribose sugar drawn. And so if we just kind of flip this around in space, we could have this red phosphate over here, and then the hydroxyl would be on this other end. And so again, I drew 5' to 3' here. As we go from the phosphate end to the end with the hydroxyl, it's 5' to 3'. I'm making a big deal out of this because this 3' hydroxyl is so important for connecting the next nucleotide. Now, I brought up the topic of Watson-Crick base pairing, and in this Watson-Crick base pairing is how they determine the structure of DNA. What they found is that anytime that you had an A on one strand of DNA, it would be matched up with a T on the second strand of a double-stranded DNA molecule. So for example, I've got an A here and a T here. Then you can have C's matched up with G's, and so I've got a C and a G matched up here. And then if T is on the top strand over here, A was on the top strand, T is on the top strand here, so A is on the bottom strand, T and A again, and then G and C. So no matter what, an A and a T and a C and a G should match up with each other. And again, that combination, A's and T's, C's and G's is Watson-Crick base pairing. The other thing that I pointed out is that there are more hydrogen bonds connecting a C and a G, there's 3 of them, than there are connecting an A and a T when they're interacting. And so there's a stronger overall strength between a C and a G than an A and a T. That'll come up later in the class, but just for now, know that there is a difference. Okay, so because this is also still kind of messy, then we can go to drawing DNA as if it were a ladder on its side. So I just drew 2 red strands, the top strand and the bottom strand. Those are the phosphate and sugar backbones. And then the rungs of the ladder are where the A's and T's, C's and G's match up. And then with the blue dots here, I was indicating where the hydrogen bonds are formed. So within this red strand, this top strand, all the phosphates, sugars, and nitrogenous bases are connected with covalent bonds, and the next nucleic acid is attached with a covalent bond. So this entire strand is made up of covalent bonds, and we know that covalent bonds are strong bonds, and then hydrogen bonds are much weaker. And so if we were to start heating up this molecule until the point where it started to come apart, it wouldn't break along the backbones. Where it's going to break is those hydrogen bonds. So here we have 2 we have a double-stranded DNA, and as we heat it up, I should have used red and then blue for cooling it down, so ignore my lack of color coordination. But when you heat it up, it's the hydrogen bonds between, say, the A on this strand and the T on this strand. Those 2 hydrogen bonds will melt, and when the 2 strands melt, then they can flow apart in the water solution that they're in. And so you go from double-stranded DNA, heat it up, it breaks right down the middle as if it's like a zipper unzipping, and then you have the 2 separate strands, which are entirely made of covalent bonds here, so they're still strong at this temperature. If you cool the 2 strands down, then the A and the T will want to match back up, and the other Watson-Crick base pairing nucleotides will want to line back up. And so when they come back to the double-stranded form, you should have the nucleotides, if this was done in a slow and controlled manner, you should have those 2 strands match back up exactly how they originally were, and it would be the sequence of the strands that align them as to how they match up, and so they will go back together exactly how they were. So that's a really interesting and important topic as you move your way up into higher levels of biology. We're going to make use of this ability to hybridize back together in the same way that they melted apart, the same way it was before it melted apart. So melting, just as if you were to melt ice, you're breaking a bunch of hydrogen bonds. You can call this melting, and so then there's going to be a certain temperature at which those 2 strands come apart, and then you cool it back down, and just like when water goes to ice, it's at the same temperature as you go from ice to water. So that, again, that same temperature, the temperature that you melted the strands, is also going to be the same temperature that they should come back together. Okay, and one final point, I wrote anti-parallel here. When I drew this top strand, I had the L shapes going from here, from the phosphate here to the sugar at the bend to the nitrogenous base. And then on the bottom strand, the phosphate and sugar backbone is running parallel to the other phosphate and sugar backbone. But the word anti comes into play because on the top strand, the 5' phosphate is on the left and the 3' hydroxyl is on the right. On the bottom, the 5' hydroxyl is on the right and the 5' phosphate's on the right and the 3' hydroxyl's on the left. And so if we draw an arrow going from the 5' to 3' direction, it runs right to left on the bottom strand, but left to right on the top strand. So they're running parallel to each other, but in opposite directions. So that's where the anti comes from. It's not that the backbones aren't parallel, it's that they are lined up backwards to each other. Okay, and that's also a really important point because when we start breaking DNA and wanting to put it back together, you're going to have the a 5' phosphate and a 3' hydroxyl are going to be used to connect back together. Okay, there's a lot of detail on that slide. Go ahead and re-listen to that if that was a little too fast and potentially unorganized. But let's go on to the next slide. And I bring this up just to show that there's a standardized way of drawing DNA. You might draw DNA horizontally, or you might draw DNA vertically. And where you put the 5' and 3' hydroxyls, there is a standard way for doing that. So it wasn't some random act that there's the top strand on the previous slide was 5' on the left and 3' on the right, and that the bottom strand was 5' on the right and 3' at the left. That's the standard way to do it. So I wouldn't want the 3' end on the top left. If you were to draw it horizontally, you would have the 5' still on the top left, and then it goes down to the 3'. Whereas on the other strand, on the right strand, it's 5' at the bottom and 3' at the top. And how I kind of remember this is from my days of leading a Cub Scout pack, and we talked about flag etiquette. If you hang a flag on the U.S. flag on the wall, the blue field with the stars should be in the top left corner when it is hanging horizontally. And then if you hang the flag vertically on the wall, you still should have the blue field on the top left. And so just like that, the RNA or DNA, double-stranded DNA, or you can have double-stranded RNA, the 5' phosphate should be on the top left. So that's just one way to both learn the standard way of drawing DNA and also double-stranded nucleic acid, and also a little flag etiquette for you today. Okay, so then on the next slide, the enzymes of the central dogma of molecular biology. Sometimes people throw molecular in, but you don't have to. So I'll just refer to it as central dogma of biology from here on out. So again, we've got DNA to RNA to protein. And in this one, I want to focus on the enzymes. So when DNA gets copied and makes more DNA, that is, you read DNA strands and make DNA strands, the enzyme that does this is called a DNA-dependent DNA polymerase. And so DNA-dependent DNA polymerase means you read DNA and then you write DNA. So if you're going to copy DNA, you need to read the DNA and write the DNA. If you're going to go from DNA to RNA, that's going to be DNA-dependent, because it's reading DNA, and then it's going to be RNA polymerase. So RNA polymerase is because it's going to write RNA. So you read the DNA and write the RNA, and the shorthand for that would be capital D, lowercase d, and capital R, lowercase p. I forgot to write it on here. But then Francis Crick noticed that you could go from RNA back to DNA, or he hypothesized that you could if there was an enzyme out there that could do the reverse of transcription. So there's reverse transcriptase, and it turns out that's true. We do have those enzymes in nature. So if you go from RNA to DNA, you're reading RNA, so it's called RNA dependent, and you're writing DNA, so it's called a DNA polymerase. And then, of course, RNA-dependent RNA polymerases are very common in RNA viruses, because the RNA viruses need to replicate their DNA. And so this would be an RNA-dependent RNA polymerase. So again, written out down here, if you're reading RNA and making RNA, it's RNA-dependent because that's what you're reading, RNA polymerase because that's what you're writing. In a paper published just last week in Nature, they increased the number of known RNA viruses by doing a deep dive data mining into a bunch of DNA sequences that have been uploaded onto the internet over the years. And for just looking for the RNA-dependent RNA polymerase, the number of known RNA viruses went up by 5-fold, which is pretty outstanding. So there's lots and lots of viruses out there to be discovered and to be categorized. And then, the the enzymes that read nucleic acids and write nucleic acids, polymerase. I keep saying polymerase, but I hadn't introduced that. And then enzymes that read nucleic acid but write protein, which is over here, is called a ribosome. So all four of these arrows on the left side of this chart are polymerase activity. The RNA to protein is translation, and that is done with a ribosome. And we're going to learn why you only have an arrow going from RNA to protein and not back in the other direction. One more slide before we get to where we left off in class on Friday. And in this case, we're going to talk about DNA replication and RNA replication. So on top, we've got DNA replication. What happens is that, again, those hydrogen bonds that are forming the double-stranded, or connecting the two strands within a double-stranded nucleic acid, are going to be the parts that will break first instead of the covalent bonds that are along the length of the strands. And so when those separate apart, you're going to have some DNA-dependent DNA polymerases come in, and they will read one strand of DNA and make the second strand. So each of these each of the black lines was single-strand DNA until the polymerase came along, made the red strand, and now it's double-stranded DNA. So you started with one double-stranded DNA and you end up with two double-stranded DNA. So that's how it is replication. The transcription looks very similar, and to be honest, I drew this a little bit wrong. I'll talk about that in a second. But we've got, just like we had a replication bubble, we also have a transcription bubble. I have the DNA-dependent RNA polymerase coming along. So here the red strand will means RNA, whereas here the red strands meant DNA because we were making a copy of DNA. So the RNA is going to be single-stranded, and I should have just drawn it coming straight off of that polymerase and not coming back onto the DNA here. But the same idea of having a bubble is still true. You're going to have this fork where you're going to be breaking as the polymerase is moving in a 5' to 3' direction. This is the 5' end of the RNA. It keeps putting a new nucleotide on the 3' end of the RNA that it's making. As it's moving in that direction, you will melt the hydrogen bonds here, will get melted or pulled apart, and then once the DNA-dependent RNA polymerase passes, the RNA strand goes off on its own, and the two strands of DNA can then go back together. So a lot of information in this lecture, and the pictures could have been drawn better, but hopefully if you missed class on Friday, me walking through these pictures in this condensed lecture was helpful. All right, again, this is Life in Biology. I'm Dr. Joel Graf, and you can always like and subscribe or not.